Alright, so let's go ahead and talk about labs. So some general rules: you need to listen or read your instructions. You need to wear safety goggles. You need to notify me if there's any spills or accidents. You need to always wash your hands. Keep your hands away from your face and tie your hair back if you have long hair. Roll those sleeves up if you have loose sleeves. Know your location of our fire extinguisher, fire blanket, eye wash station, and first aid kit. Keep your area clean. If you have contact lenses, it's actually better to wear glasses, but it is not required. Okay, I wear contact lenses. Never put anything in your mouth. Clean up your area when you are finished, and never, ever, ever horseplay or practical jokes in our lab. So we are going to deal with some glassware. If it's cracked or chipped, please notify me because I need to place it in a special glass container. Pour liquids in. When you are pouring liquids into glassware, please make sure that the container you're pouring in is actually on the table. So if we have a stirring rod that's available, please put down a stirring rod to prevent liquids from splashing. Okay, so if your glassware does get broken, please notify me so I can clean it up. And you're not going to have to worry about glass tubing. Now, same thing with, we're not going to worry about hot glassware because we are not going to use hot glassware in biology. Now, chemicals, again, so if I have to wear the goofy goggles, you have to wear the goofy goggles. Okay, so never, ever, ever mix together your chemicals unless I tell you to do so. And that also goes for never, ever, ever taste any of your chemicals. You should never be eating anything in a lab unless you are instructed to do so. Same thing with smelling, okay? If you're going to smell an odor, you need to waft it. Basically, you're going to waft the scent towards your nose. You're not going to directly put your nose over the container and inhale, okay? Never pour water into your... All right, so let's look at some chemical safety, okay? So if you need to follow all instructions on how I want you to dispose of the chemical, okay? Sometimes I might say, hey, you can just put it down the sink, or hey, you need to put it in this special container, okay? So again, always, 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 always wash your hands. That is very, very... Alright, so you need to always pull the plug, not the cord, and make sure that you do unplug all electrical equipment at the end of... So if you do get a cut or a bruise, please don't ever touch it or let anybody else touch it, that open wound without safety gloves, okay? So don't let anybody touch it, period. Make sure that you notify me so I can actually put the appropriate, either if we need bandages or if we need some additional first aid. So if somebody does faint, don't everybody rush in to save them, okay? That's actually the worst thing. They need fresh air in order to recover from that fainting spell. So if you do get something in your eyes, okay, you want to make sure that you flush them. We do have a eye wash station, okay? If a foreign object is getting lodged in your eye, please don't ever rub it, okay? Because then you can damage your cornea. So if you drink something that you weren't supposed to be drinking, okay, you need to tell me because I need to see what did you consume, and then I need to call the nurse and possibly the poison control. So if you do spill something on yourself, okay, make sure first thing before you even think about running to the sink, please notify me, because if it is an acid, I need to put a base on it. If it's a base, I need to put an acid on it in order to buff it, okay? So again, if you have acid and you put water on it, it actually causes this really bad chemical reaction. So if you get electrocuted, okay, I don't know why you would, but you never know, right? Okay, I'm going to shut off the power, and we're going to remove it. We're going to remove that wire, okay, with rubber gloves. So please, please, please tell me. Alright, so in biology, which is the study of life, okay, we are going to look at 7 characteristics. So I want you to take a minute, and I want you to think about what defines life. So 7 characteristics. The first one is all living things are made out of cells. Okay, so each living organism shows order. Okay, so the smallest is cells. When you have a bunch of cells. So the cilia, okay, we also have cilia in our trachea or our windpipe. If we do a cross section of the cilium through using our microscope, okay, it's actually identical order. Okay, they're both made out of cells, which therefore is. So our second one is all living things respond, okay, to our environment. They respond to change, which a change is called a stimuli. So if you look at this, we've got a fly that's being eaten by a Venus flytrap. Okay, the stimulus for the Venus flytrap was the movement of the fly, and its response is to close its appendages. So all living things need to maintain internal balance, okay, or maintain homeostasis or sameness, okay. Believe it or not, if you get overheated, you sweat, okay. That is how your body is trying to cool you down or lower your body temperature back to 98.7. Well, chickens don't have that ability, okay. Chickens cannot sweat. They actually pant like dogs. Number 4, all living things must reproduce. Okay, in order to reproduce, you must be able to pass your genes along to healthy, fertile offsprings. Fertile offsprings means you have to be able to mate. Okay. All living things must be able to grow, okay? So they grow based off of a sequence, okay, based off of instructions that are presented in their genes, okay, or their DNA. So if we look at this frog, the young frog, it takes about 84 days for the transformation or metamorphosis. Now our last one, all living things are made of the universal genetic code or DNA. Okay, so DNA controls your hair color. So let's look at our organization. Okay, so starting from the greatest is a biosphere or planet Earth. When we go to our next level, this is an ecosystem. An ecosystem has non-living and living factors. Non-living means abiotic, means not alive, and biotic is life, because bio for our prefix for biology means life, and ology means study of. So we're going to look at, for an ecosystem, we're looking at non-living things such as this rock or the water or the climate or the weather or the soil. And we're also going to look at the various animals that are involved in plants. Now the community is all these various of populations. So we have a buzzard, we have wildebeest, we have lions, we have giraffes, we have rhinoceros, elephants, we have the various types of trees. They're all interacting amongst each other. Now our next lower unit is a population. Population is only one type of species. Now, an organism is one individual. So when we So again, even deeper into cells, we have molecules. So molecules are groups of atoms. Then we have cells. Then we have groups of cells such as tissues, organs, organ systems, and an organism. So then we have, again, populations. That's one particular animal or one particular species. Community is all of those living together, all those various populations. Ecosystem is living with non-living things, and biosphere is our planet Earth. All right, so some fields of study that you can possibly study if you decide to go into the biology field. Okay, so cell biology, you're studying cells like this white blood cell. Okay, so taxonomy is the study of naming and how things are connected. Evolution, like how things present day, so here are present day finches, and how did they come to be? Okay, so their ancestors, how did they? So let's do a little check. Okay, so how many characteristics of life are there? There are 7. What is the smallest unit of life? Cells. All living things must be able to blank or to make one of their own kind. So think about those 7 characteristics. Reproduce. All living things have a blank, which are like instructions for building the body. Genetic code or DNA. Number 5, during development, some cells become liver cells, some become skin cells, and some become muscle cells. This process is called blank. This is actually called cell specialization, okay, or where the cell therefore becomes tissues, okay, or they have a special job, okay. So each of these guys, the liver, skin, and muscles, they all have specific jobs or specific functions. Number 6, animals get their blank by consuming food. ATP or energy, adenine triphosphate. Okay, plants get it by photosynthesis or by making their own food. Number 7, blank is maintaining of an internal balance or sameness. This is homeostasis. Number 8, blank is the process where species change over time. Evolution. All right, so let's talk about the goals. Okay? Natural world. So our first one is our first step. Okay, so we have to observe or go through. Science always begins with observations. Okay, so what does it mean to observe? Okay, observe means that you notice things and you describe events in a process that's very organized. So I want you to try this exercise. Okay, how well is your organization or your observing skills? Okay, I want you to find Waldo. Okay, if you've never seen the book, okay, or have ever seen these illustrations, Waldo is a young man that has a red and white striped shirt and a red and white striped hat. So take a minute or two and see if you can find Waldo. So Waldo is right next to his grandmother right here. But you might have got tricked. So let's think about it, okay? So where did science come to be, okay? So we didn't have scientists, we had philosophers, okay? So philosophers actually didn't really conduct use logic instead. Okay, so with his with his theory, okay, we have to then through our scientific investigation remain the same. So if we look after our experiment, we have to collect our data. Now there's two different types. There's quantitative with an N, okay? That N is for numbers, okay? So this is, if we're looking at our monkeys, there are 3 monkeys. But if we're looking at quadrants, so Reddy came along and he wanted to actually have an experiment to test this spontaneous generation that Aristotle suggested. Okay, so he took a flask that was unsealed with meat. He took another jar, he sealed it, and then another jar he covered with gauze. Okay, so this very first one is the control. Used. Now, these 2 additional flasks, okay, we call them the experimental group. Okay, the reason why is so only the unsealed flask grew the maggots. Okay, so Reddy's conclusion stated that his hypothesis was correct or was supported. Okay, so maggots did not spontaneously generated because we know that flies actually are the ones that land and produce those maggots or those baby flies. So the hypothesis. So some errors, okay? Every experiment, you're going to have errors. You always want to write down at least 3 errors. Okay, probability is the likelihood of something happening, okay, and it's simply due to random chance, okay? So the effect of random chance are countered with having a large sample size, okay? So instead of doing it all to chance, we want it very organized. We want to make sure that it wasn't due to random chance, okay? So therefore, we can actually use a large sample size. So in our ready experiment, now, scientific method. Okay, so again, we got to get our question, we got to make our observation, we got to gather information, we have to form the hypothesis. It has to be if the IV, then the DV. And we have to set up our controlled experiment. So again, what I want you to do is take a minute. Okay, so again, depending on your results, your hypothesis might be supported, might be rejected. If it's rejected, you need to modify it, okay? And then you need to adjust your experiment, okay? So again, you need to design, adjust, modify your hypothesis, modify your experiment, and conduct some until basically so again, after you have done this and you have found no errors, okay, and you're okay, it can be part of our scientific understanding. So again, what is the difference between a hypothesis and a theory? What goes up? So graphing. Okay, so we have a couple different steps that you need, and hopefully you've learned this in math. Fingers crossed. If not, we're going to review it. Okay, so you need to decide on the graph type. So line is for continuous data, bar is for category, pi is for percentages. Okay, you need to label your x and y. Make sure you include those units. Determine your scale. Okay, so your scale should be as large as possible within the limits of your paper. Okay, so example, you can have multiples that might be easier to work with. 1s, 2s, 5s, 10s, 25s. Then you need a plot. Okay, you need to plot the data. You need to include a key or a legend, okay, especially if there's one or more sets of data. You need to connect those points if you're using a line graph. And if you're not using a line graph, you need to, and you're using a scatter plot, you need to draw a best fit line. A best fit line is a line that goes through the majority or the average of your data points. Then you need to title it, okay? Always include, okay? The title is always set up this way. The effect of the IV on the DV. Always, always, always. So I want you to take a minute and I want you to